Hi there, friends. My name is Peregrine. I am really excited to welcome you back to Lab at Home, and I've got someone really special with me today. Kind of before we introduce her, I wanted to let you all know that we hope, as always, that you'll let us know what you're thinking and about any questions that you might have in the chat box, which our Catalyst Volunteer Caroline is moderating for us today. So I hope all of you will help me welcome our scientist spotlight guest here, Allie Schrock, who is a scientist who works with the Drea Lab at Duke University in the Department of Evolutionary Anthropology. I think she can probably do a better job of introducing herself than I can, so I'm going to go ahead and hand it over to her.
Thanks, Peregrine. Yeah, I'm super excited to be here today. My name is Allie, and yeah, like Peregrine said, I'm a researcher at Duke University and a PhD student there, and I study lemurs and their behavior and their brains, which we are going to talk some about today.
That's so cool. All right, well, friends, but before we begin, let me go ahead and say that today we are going to get the chance to see some actual real live, well, not live, but some actual lemur brains. Allie will also show us how she studies them, which involves taking like a paper thin slice of the brain. We will actually get to see her do that on this program. I'm really excited to see what that looks like. We'll talk more about the specifics in just a few minutes, but I wanted to give you all that heads up, no pun intended. So I know that I have a lot of questions for you, Allie, and I hope that some of our friends will think about what they would like to ask a lemur brain scientist as well. But I guess I would start in asking, you're a research scientist, and can you tell us exactly what it is that you study?
Yeah, so I'm mostly interested in how animals' brains and their hormones, so those are different things that go throughout your body, are related to how they behave. So for the most part, I study this in non-human primates, which includes monkeys, apes, and of course, currently lemurs. So yeah, I just study animal behavior and how that relates to the brain and some different hormones.
That's really cool. What got you interested in that area?
It's a great question. I guess I can take it way back. I actually grew up in the Durham area, and I have always loved animals. I love, dogs, cats, everything. When I was a kid, I actually took a Girl Scout outing to the Duke Lemur Center, and I got to see that. I loved going to the zoo, loved, just all that sort of thing. And so I figured always oh, I'll be a vet, because that is what you do. If you love animals, you become a vet. But as I got a little older, I kind of thought that maybe that's not what I wanted to do. I don't like to see animals in pain or sick. And so I kind of decided that maybe being a vet wasn't for me. So I didn't really know what I would do. But when I was looking into colleges, I learned about Bucknell University, which is in Pennsylvania. And there they actually have a major in animal behavior. And so when I was reading about it, I read about, this whole field of animal behavior. And so I just thought, that's really what I want to do. It sounded so interesting, so amazing. So that's what I did. I went to Bucknell University for college, and there they actually have some species of monkeys on campus that students can study. So I went to college, and I got to study capuchin monkeys and their social behavior. And so that's really what got me into studying monkey behavior or primate behavior. And from there, I got to learn about their behavior, and I was really interested in kind of some of the underlying mechanisms or things that might be steering this behavior within the body and the brain. So that's kind of what led me to where I am today. And so now I study behavior and the brain.
That sounds like the perfect duo. If you want to study animals and why they do the things that they do and how they do the things that they do. We have a really great question from one of our friends asking, Allie, where are you right now?
Right now, I am actually in our lab space on campus at Duke University here in Durham. So we don't have lemurs on campus. The lemurs are at the Duke Lemur Center, but here in the lab on campus is where we do some of our different studying. This is where I look at the brains, and other members of the Dre Lab do their studies. So I'm here in the lab on campus.
That's so cool to get an exclusive look. I'm so excited to kind of see the stuff that you have to show us. I guess I want to kind of dive into the science a little bit. So Allie studies lemur brains and their behavior. Before we ask our expert, I wanted to ask all of you, what does a brain, like any brain, have to do with behavior, with the way that something acts, like whether it's a lemur or a person or like anything with a brain? What does a brain do? Let me know in the chat what you're thinking. What does the brain do? Because I know Allie is going to have some really cool stuff to tell us about. We have a friend in the chat saying that the brain controls the body. That's a great thought. So the brain controls the body. Anything else that it does? Or maybe do we know how it does that? What do you think? Or even like kind of what comes to mind when we're talking about it? No pun intended. Ooh, nerves. I love this. That's a great word associated with behavior in the brain. That's great. We say that the brain works. It works super hard. We need our brain to help us do just about everything.
I better hope your brain works.
Exactly. Well, I would love to keep hearing what you all are thinking, but I'd love to turn this question to you, Allie. What does a lemur's brain have to do with a lemur's behavior?
Yeah, it's a great question. And kind of one thing that I think it helps to think about is that different areas of the brain are responsible for different functions in the body. So I can show you a little bit of an example of lemur brain, if that sounds good. And we can look at some of the different areas and what they do. So now what I'm going to do is I'm going to flip my camera, and here we are looking inside of the machine called the cryostat machine, where I work on the brains. So the brains are frozen, so they have to be really, really cold. So I have to look at them inside of this cryostat machine. So first, I'm going to show you this brain. This is from a lemur called a Schrock. And I don't know if any of you have ever watched Zebumafu, but Zebumafu was a Schrock. So that is the type of lemur that this brain actually came from. The brains are from the Duke Lemur Center, from animals who lived a long, very happy life there. And they died of natural causes or old age. And now we get to continue learning from them by studying their brains. So this is a Schrock brain, and I'm not going to touch it too much because the brain has to stay really cold and my hands are warm. But one thing is on the front of the brain here. So this is the front. And in lemurs, this one area in the front of the brain, it's called the olfactory bulb. And that's really important for lemurs because olfaction means smell. And so for lemurs, smell is really important in their life. They use it to know who their friends are, where their territory is. And so that's one thing that lemurs have. Lemurs have a really large olfactory bulb compared to their brain size. We actually don't even have the whole thing here because it protrudes pretty far and it's harder to get out. So that's one area of a lemur's brain that kind of determines its behavior. So it has this big olfactory bulb that lets it smell, so that way it can know who its friends are and things like that. Another brain area that we might be able to talk about is back here in the back of the brain. This is called the cerebellum. And you might have heard of it because the cerebellum is actually responsible for a lot of movement or motion. So in this area of the brain, it really controls your balance, your movement, and things like that. So someone had said that the brain has nerves. So yeah, nerves are actually all throughout your body, and they connect to the brain and help your body do its different functions. So a lot of the nerves are important for the cerebellum to be able to help your body do these different things that it does. So
we're saying that we also have a cerebellum, is that right?
Yes. Lemurs are actually primates, just like humans are primates. And so a lot of things about our brains are similar. So yeah, humans have a cerebellum. Humans have an olfactory bulb. It's smaller in comparison to our brain size than a lemur's, but we have a lot of the same brain areas as a lemur.
That is so cool. We have someone in the chat asking if you feel a little bit nervous handling a lemur brain. What do you think?
Yeah, sometimes I do, especially when I just want to make sure that, like I said, the lemur brains stay really cold. I sometimes wake up at night and worry that I left a lemur brain out of the freezer, and that it's melting and I won't be able to use it because, like I said, we need them to stay really cold in order to study them. And it looks like someone asked, why do the brains have to stay really cold? And that's a great question. So along with just looking at the brain and just how the different anatomy and how it looks, one thing that I'm interested in studying in the brain is different hormone receptors. So hormones are these substances that go throughout the body, and they can interact in lots of different places, including the brain. And in order to have their effects, hormones have to have receptors. And hormone receptors, basically, they need to stay really cold in order for those receptors to stay where they were when the animal was living. Because if they're not cold, then the molecules might kind of start to degrade and we won't be able to see them anymore when we look for them. So that's one of the main reasons that we have to keep our brains very cold. Yeah.
So it needs to be a brainsicle the whole time.
Yes, the whole time. One other thing about this, I guess, if you're looking at it, this is my hand, just to kind of show you a size comparison. This brain is probably about the size of maybe like a lime, if you wanted to think about that. And so Schrock are some of the bigger lemurs that we can look at. But I also have another brain that is from a mouse lemur. So a mouse lemur is very small. It's about the size of a mouse. And this is a mouse lemur brain. So you can see it's much smaller than a Schrock brain, which makes sense because a mouse lemur is much smaller than a Schrock. So they don't need as big of a brain.
Wow. So is it about equivalent? Like, is a mouse lemur's brain smaller in a mouse lemur's head than a Schrock lemur brain is in a Schrock lemur head, or is it about the same?
Honestly, I don't know the exact numbers, but I imagine that it's about the same. So we see differences in relative brain size, relative, the Schrock brain to the body size, and things like that. We see differences in relative brain size between like lemurs and monkeys, or monkeys and humans. But within lemurs, it's more similar, if that makes sense.
It does, yeah. What do you think, Friends? Is there anything that you notice about these really amazing specimen, or any other questions that you might have before we kind of talk more about the method that Allie uses to study these brains? What do you think? So, yeah, I noticed that it has some wrinkling on the top. It looks like that cerebellum area is a little bit kind of more wrinkly, but that there are some other wrinkles. We have someone who said they noticed some little red lines on there. What would those be?
Yeah, so starting with the little red lines, those are probably remnants of, they could be nerves and different cells. Some of the red could also be remnants of the vascular system, so the blood that goes around. The brain needs blood just as much as other parts of the body. So that's kind of some remnants from that potentially. And then as far as the wrinkles on the brain, that's a great observation. So different species have different amounts of wrinkling on their brain. And the purpose of those wrinkles is to increase the surface area in the brain. So if you have more surface area in your brain, you can have more brain cells and do more functions, have more processing power, be able to think more, think harder, things like that. So yeah, so you can see some folding on this brain. It's harder to see on the mouse lemur brain because of how it was prepared. But yeah, so there's definitely some folding on the brain. And for example, if we were looking at a human brain, we would see a lot more folding a lot more wrinkling. So it just varies between species and between what type of animal you're looking at.
So it's kind of like if we're thinking about like a piece of paper and like crumpling it all up, it's still the same size as a piece of paper. It's just kind of folded in on itself.
Yes, right.
That's so interesting. I know that we have a friend who's asking, is it fun to study these brains? What do you think?
You know, on different days, I would probably have different answers to that question. It can be really fun, and I think it's so interesting, and it's really exciting because one of the reasons that I'm studying these brains is actually because we don't know a lot about lemur brains, and so I think it's very exciting to be able to study this new thing that we don't know a lot about. But sometimes it can be frustrating as well, as with most things, I think. I'm probably going to show you in a minute how I slice these brains, and sometimes my big slicer machine here does not want to cooperate with me very nicely. And so sometimes it can be frustrating. Sometimes it's just challenging, doing the work. But overall, I would say yes. I think it's fun and exciting. Otherwise, I probably wouldn't be doing it.
I'm so glad that it's fun for you, and I appreciate you telling us too that sometimes it can be frustrating, just like anything in life can be, and we're really happy that it's something that even when it gets frustrating, you love enough to keep doing. We have another really good question from a friend in the chat saying, is the brain hard or soft?
That is a great question. So these ones, they are hard. I can pick it up and kind of tap on it. It's hard because it's, like I said, it's frozen, very cold. If this brain were not frozen, it would be very, very soft. For example, if the brain wasn't frozen and it had just, come out of the lemur, it has a consistency more like Jello. Like I don't want to get too graphic here, but it's very soft if it's not frozen. But like I said, we need it frozen in order to look at what we want to look at. So they're frozen, and yeah, they're hard, not soft.
That makes sense. I don't know if it's too kind of round on top, but would it be possible for us to take a look at the underside of the brain as well?
Yeah. So this is what the bottom of the brain looks like. So here we can kind of see some different nerves. And you'll notice this is more of the olfactory bulb that I was mentioning earlier. But it actually is pretty flat on the bottom, and that's not natural. That's not how it would normally look inside a lemur. But this is just due to the way that it was frozen. So like I said, when the brain comes out, it's very soft. And then normally at the lemur center, they will lay the brain on the bottom, and then it will kind of just freeze flat like this.
Wow. We have a couple really cool questions like that. First, we have someone asking, do you only dissect the brain, or do you dissect any other organs or body parts of the lemurs?
That's a good question. For me, I only dissect brains. I only study brains, but there are lots of other scientists who do study different organs and different parts of lemurs for whatever reasons that they might be interested in. But I only study brains.
Sounds like there's a lot to study, so that makes sense to me.
Yes.
And we have some questions kind of about maybe some of the pieces we're seeing. We have someone asking, are the white bits we're seeing kind of where the spine connects? Are we seeing bones? We have a question about kind of what the peanut-like shaped thing is, kind of at that central area of the brain, I think.
Okay, I can answer a few of those. So there are no bones. The brain is encased in the skull. So those would be the skull would be all around the brain. So there's no bones that we're looking at here. The spinal column would connect actually back here. So this region is known as the brain stem back here. So humans, like, we also have a brain stem. And so that is kind of where the spinal column connects to the brain, and some of those other nerves are. And I'm not entirely sure what the peanut structure would be referring to.
It's maybe that kind of oval, almost kind of more tan.
Right here.
Maybe that one.
I'm not entirely sure. It actually is a lot easier to view the brain anatomy when we cut the brain. And so probably someone could identify that structure from the external anatomy, but I am not one of those people. So I'm not entirely sure exactly what that region would be. But like I said, that is one of the reasons why we cut the brains and don't just study them whole, so we can see some of the internal structures.
You're kind of more used to looking at it from the cross section. We have a couple of friends
Right.
who are really interested in seeing what it looks like when you cut the brain. Is that something we might be able to take a look at?
Yeah, we can go ahead and look at that. Let me just move these two brains out of the way.
Those are it was so cool to see that.
Yeah, so now I can go ahead and cut the brain. So this is a brain that I have been working on cutting. This is a ring-tailed lemur brain, actually. And so you can see this is in my machine, and we have the brain mounted on this little disc to hold it in here. And then what I'm going to do is I'm going to put this little glass piece down, and that helps the brain to not roll up once I cut it, because I'm going to cut it into a very thin section using, I should show the blade. So this is a blade right here. It's very, very sharp, like a very sharp knife. So that's what's going to make the actual cut. And then this glass plate is going to hopefully stop the brain section from rolling up. So I have a little wheel on the side of the machine here that you can't see, but I'm turning it. And now we have a brain section. So one of the frustrating things that happens sometimes is when it sticks to the glass. So I'm going to try to take it off here. And we use these paintbrushes to kind of manipulate the brain sections because they're so thin that you can't, you have to be very delicate with them. So
it happened so quickly. I was expecting it to be sort of a slow, careful slice, but it was really very fast.
Yeah, there are different methods for doing it. Sometimes people have different ways of doing it that are slower, but because our brains are frozen and for what we're doing with them, we can cut them pretty quick. So that's what I did. And then I don't like it to be wrinkly because then it won't be able to go onto the slide that I'm going to mount it on as easily. So I'll try to tap out some of the folds and wrinkles that we have. Sometimes it goes better than others.
Mm-hmm.
And
it
then
is so paper thin. You even said, I think you said earlier, kind of how thin you cut it. Could you tell us again kind of how thick that is?
Yeah, so each slice is 20 micrometers thick. So, 1 meter is about 3 feet. You might think of a centimeter. There's how many centimeters are in a foot? Like 30 or something. But so basically this is a very, very thin slice. If you were to count how many of these would fit in an inch, it's over 1,000. So there's over 1,000 slices in 1 inch. So it's very, very thin. Oh no, it's gonna stick here.
We have someone asking, is the texture kind of like paper as well?
It's similar, but as you can probably see, it's very, like I said, it's very delicate, and it can get kind of ripped or damaged very easily. And so now that I've somewhat got my section all flattened out, I'm going to take a slide. So this is what we use. This is like a microscope slide. So you could put lots of different things on this to look at them under a microscope. We actually don't look at these under a microscope, but we use microscope slides. And what I'm going to do is just kind of roll this on. That didn't go great. But yeah, so you can see there's a little bit of a fold and a bubble on there, which we don't usually want. But you can kind of see what it looks like once it's mounted onto the slide. So it gets more transparent once it goes onto this glass slide. And then this is how we use to study. So we have different techniques that we can use to stain the brain so that the different cells will get stained. We can see different areas of the brain, and then also use different techniques to look for the hormone receptors like I was talking about before.
Wow, that is so cool. I didn't even realize, right, when you first cut it, it kind of looked like opaque paper, but yeah, exactly, when you put it on that slide, you can almost see right through it.
Yes.
That is so cool. So it's
Yeah.
something that you don't look at under a microscope usually, you said?
Yeah, so we don't necessarily look at these under a microscope. At least that's not in our current procedure, but maybe in the future we might decide that that's something that we would want to do. But yeah, so we actually, like I said, we stain the slides. So that kind of involves basically just dunking them in various different chemicals and substances to be able to look at the different structures. And then we also do kind of a similar procedure in order to be able to view the hormone receptors.
Wow, that's so amazing that you're able to kind of see something that controls actions in a way that's so so physical, and something that you can stain and see all of the differences in, kind of right in front of you. It's so interesting to think that the ways that these animals are acting is connected to this body part. It's kind of I don't know about you guys, it's kind of a weird thing for me to think about, but it's really, really amazing. Thank you for showing us all of that, Allie.
Yeah, of course.
Could I ask you, maybe we have time for maybe one last question for me, and then we'll see if there's any other questions from any of our other friends.
Sure. Here, I can switch my view back.
Awesome. So I wanted to ask, you told us about you told us about things that can be a little bit frustrating, and I wanted to ask too about what is your favorite thing about being a scientist and studying what you study? What makes you the most happy?
It's another good question. I think the things that I like about it are thankfully more than things that are frustrating. But I would say just one really fun story that I can think of is actually from the Duke Lemur Center. So in addition to studying brains, I also study behavior and cognition in the living animals. There was one study that we were doing where the animals were basically presented with a choice of 3 cups, and there's a treat under one cup, and the animal had to pick which one. They basically had to remember where the treat was between the 3 cups. And lemurs are very interesting because they don't use their hands a lot. They are primates, and so they have hands and opposable thumbs and everything, but they're much more likely to use their nose to do things. So it was really funny to watch the lemurs point at the different cups with their nose. And my personal favorite was one lemur who would choose by sticking out their tongue. So the lemur would look at which cup they thought the treat was under, and then they would go and stick out their tongue and point at it. And it was just so funny to watch. And so I think it's just fun things like that, that we're learning about the animals, and also just getting to kind of see them do their natural behaviors, and just have a lot of fun, studying animals, which is what I always wanted to do.
That's so cool, and that's so cute to envision. I like that it also shows you that even though they have similarities in behavior, they're also individuals. So it was funny to see that. That sounds really, really amazing. I want to thank you so much for being here, Allie. I hope that my friends in the chat can join us in thanking Allie for taking the time to show us the lab, and even to see a slice of those brains and the brains themselves. I loved seeing the difference between the Schrock and the mouse lemur brain. That was so amazing.
Yeah, thank you for having me. It's been great.
We really, really enjoyed it. I did, especially because I know that I really, really enjoy lemurs and primates and stuff like that. So I really liked that. And I look forward to having more scientist spotlights where we can meet more scientists doing science, especially in our area. I hope that You might join us, friends, next week where we will have Little Lab. We're going to be looking at ocean currents. And then the week after that, we are going to kick off Black History Month by studying one of my favorite historical Black biologists. And we're going to be making our own microscope for that as well. So I hope that we will see you next week and maybe the week after. Allie, thank you again so much for being here. I think we'll go ahead and wave goodbye. Thanks so much, friends.